Hello and welcome to Learn Extra Live. This is Aslam and I'm Katlaho. How are you, Aslam?
Great. Ready to go,
Ready
Kalleho.
to go. Hey, what are we doing today?
Today we are doing a bit of everything. Paper
Okay.
1 and paper 2.
Okay.
We're trying to cover some of the stuff we didn't cover in the last couple of shows.
Okay, that's awesome. That's great. Thank you very much. So you guys heard that we are doing everything today, grade 12s, because it is so close to your finals. Some of you have just finished prelims and you're thinking, oh my goodness, there's so much more to cover. So that is why we are here. And I also want to remind you about the spring school happening in 6 days time now. So in 6 days, on the 1st of October, we have some more spring school for you because I know you guys are going to rock at those exams. But for today, if you have any questions, please don't hesitate to ask us. Facebook.com/learnextra and our Twitter handle. Make sure that today you are on your absolute T because it is so important to know this stuff, guys. We're doing everything today. So today you're open to ask whatever you need to ask. Over to you, Aslam.
Thanks, Katleho. Welcome, mindsetters. And let's spring into spring with all the revision that we need to do. We're going to take a few questions that we have, but please, guys, from your side, whatever is bothering you, be it paper 1, be it paper 2 stuff, be it the skills or drawing graphs or whatever it is, whatever is bothering you, post those questions or call us. We will deal with your questions. We're taking off from where we were last week in terms of the year, and we felt it was important to round up some of the stuff on this diagram that makes it easier for you to be able to learn your work. And the question says, study the diagram below and answer the questions that follow. Gives you a schematic diagram of the inner ear. Now remember, when you see the cochlea, you see it as like a snail's shell in a model or in a diagram as such. This one here, we're stretching those canals out longitudinally that way. There's your vestibular canal there. I should be saying not yours, but the vestibular canal. Vestibular canal. And this one here would be the tympanic canal. And this would be the middle canal. So the first thing in a diagram like this, map yourself. Find out, use your GPS in your brains to find out where you are. First of all, we're dealing with the ear. They tell you that immediately. And you look at all the clues and you label these things quickly in pencil on your question paper. They say, explain, the first question, explain how having a severe cold, how a severe cold could affect a person who dives to great depths in the water. Now, a little bit of science in this question. Diving to great depths, as you are going deeper, pressure increases on the outside. Pressure decreases with altitude and increases with depth. As you go down, the pressure gets greater. So that's where the problem is. Now here, they're adding a problem. They're saying that the person has a severe cold. Now, if you have a severe cold, you would know that your nasal passage would be blocked. And sometimes you feel block in your ear as well, Katleho.
Yes.
Yes? So your sinuses are blocked. And in the ear, because we're dealing with the ear, our answer must be more related to the ear. Because they wouldn't put this diagram here and then ask you a question about the fingers. They're asking you about the ear, obviously. So they want to know what will happen there. Most probably, in a question like this, you look at the function of the eustachian tube. Remember, the eustachian tube is continuous with the pharynx and helps to maintain a balance on either side of the eardrum, so that the pressure on either side of the eardrum is equal. If it is greater on one of the two sides, it will pop in the opposite direction. So in this case here, if a person has a severe cold, then obviously he or she would have blockage in the eustachian tube. And because of this, it would be difficult to maintain equal pressure on either side of the eardrum. Now, this could be dangerous because if any one of the two sides of pressure is too high or too low, the eardrum would burst. Also, as you're going down, you are going to feel pain in this case. Remember, we discussed questions on middle ear infection. Same thing, because there's liquid buildup in the air-filled cavity of the middle ear, this causes pain when it pushes against the eardrum. So that's where we have problems. So this is not a great idea if you have a cold or a flu to go diving. Next question, explain how the process of hearing may be affected if part A is damaged. Part A, what is this? Look at where is it coming from. It's coming from the organ of Corti. You can see the branches. This would be the auditory nerve. By the way, the auditory nerve is an example of a sensory neuron. That means it transmits impulses from the receptor being the organ of Corti, to the cerebral cortex, the cortex of the cerebrum of the brain, where it would be interpreted. So obviously, and I've been saying this all along, when they ask you questions like these, where something is damaged, what are they actually asking you? They're asking to see whether you know the function of that part. So this is what I asked you earlier. What is the function? We labeled it first. A is the auditory nerve. We then looked at its function. So obviously, if it is not there, then that function will not take place. So in this case, we would say then that if A is damaged, then the impulse would not reach the cerebrum, and therefore would not be interpreted. That's what we're talking about there. Straightforward. We remember, in any predict or explain, this is when something is missing. Whenever you get a question like this, you ask yourself, what is this part that they're talking about? What is its function? If its function is not, if the part is not there, then that function will not carry out. As straightforward as that. And this question is why I put this question back here, this last question. Explain how we hear using this diagram. Because this diagram helps you to understand how hearing occurs in the inner ear. From the outside, it's very easy. The pinna traps the sound waves, directs these sound waves into the auditory canal. These vibrations then beat onto the eardrum, which in turn beats onto the hammer, hammer, anvil, anvil, stirrup, stirrup, oval window. Up to that point, most learners would get it right. They would get full marks if we had to ask that question. But once we go to the inner ear, confusion starts rambling around. So that's why a diagram like this is great to make you understand what is happening there. Look at where we stopped. There was the eardrum. This would be hammer, anvil, stirrup. There's the stirrup. Here's the oval window there. So what would happen here is that the vibration would start there. I labeled this earlier as the vestibular canal, and this as the tympanic canal, and this as the middle canal. We know that in the outer two canals, we have the fluid called perilymph. And inside, we have endolymph. Now, if there's a vibration on the oval window here, then this vibration would be passed on inside here, through there. In other words, all these things will vibrate. And remember that this at the bottom is a membrane, and that's another membrane. That one there is the Reisner's membrane. And this one at the bottom, the basilar membrane. So what would happen then? If the fluid is vibrating, then obviously these two membranes would also vibrate. And if they vibrate, then the endolymph on the inside here would also start vibrating. And if that happens, the hair cells that are lying on the basilar membrane will vibrate. And when the hair cells touch the tectoral membrane that's above it, the stimulus is converted to an impulse. From there, it goes to the auditory nerve. From the auditory nerve to the brain, where? To the cerebral cortex, where it is interpreted. Now, I want to show you how easy it is to explain that. Just what I'm suffering with this extending the page today. Okay, so what we're saying is that the oval window vibrates. What's the points here, please? That's your first point. What does this do? It causes vibrations in the perilymph of vestibular and tympanic canals. Step by step, when you're answering. What does this do? It causes a Reisner and basilar membrane to vibrate. Let's put the 2 there. What does this do? Causes the endolymph of the middle canal to vibrate. And because of this, the hair cells vibrate. When the hair cells touch the tectoral membrane, the stimulus is converted to an impulse. And this impulse is going to go to the brain. And that is how we hear people. So look at the set out. Now, if you look at a memo, any memo, pick up a memo from a past paper, you will see that the memo is set out in this way. And this is how the marker is going to see the answer. And if you present your answer in this way, you would be able to get full credit in any of the answers that you give. Next question, 1.14, explain how the ear helps us to maintain balance. Now we are not talking about hearing, we are talking about balance. And there are two parts of the ear, two sets of parts in the ear that help us to do this. One is the semicircular canals, and two, the sacculus and utriculus. These are the two important areas which help us with balance. Remember, the one function of the ear is hearing. The next function of the ear is balance. So the semicircular canals, and I'm not going to go into too much of detail here, the semicircular canals informs us about the direction and the speed at which we are moving. So we can move in any direction. We know we're moving sideways, side to the left, to the front, to the back. We know this. And I'm moving slowly. And I'm moving faster now. Can you see what we're saying? So the semicircular canals inform us about this. How do they do this? They have fluid into in their tubes that will sense this. And in the fluid, they have sensory cells. The semicircular canals, notice the sound of the C coming out. Semicircular canals, I would like to say it that way. They have what we call the cupula. Notice I said the C comes out strongly. And the cupula is a coat of jelly with some hair on it. It's a sensory receptor of balance. And the sacculus and the utriculus have macula. If you want to, that is S-U-M. Sacculus, utriculus have maculae, some. Semicircular canals, cupula, the C sound. Sacculus and utriculus, maculae. The maculae also have jelly and hair, but in addition to this, they have calcium carbonate crystals, which are sensitive to gravity. And because they are sensitive to gravity, they will settle differently when they are exposed to different stimuli. And, for example, if I'm standing upright, my head is straight forward. So all my maculae are settling down evenly. So they evenly sensitized. So I know at this stage my head is upright. But the minute I do this, I already know that, you know what, my head is tilted to the right, or to the left, or back, or front, or if I take it right around. All these things are possible because different maculae are being sensitized because of the gravity. If I'm moving it that way, then obviously all moving this way. So these ones are being sensitized when the fluid moves. When I move it that way, it's going to be the same thing. And if I had to go down on my head and do that, then too, I know that I am upside down, Katlego.
Yeah,
Can you do
wow. I can't actually. I'm not that strong,
that?
actually.
Yes. And you don't have to be strong to that, Katlego.
No, but you have to have some sort of balance, right?
You have to have balance. That's
That's
the
it.
thing. And when I was in standard 2, that would be grade 4 in today's time, right?
Yes.
My teacher would pinch me all over my arms if it wasn't straight.
Seriously?
And to this day, I can do the headstand anyway. I challenge anybody. I can do the headstand on that there. I'm not going to do it today because we got a show to finish, right?
Yeah, no, okay. Let's have
Okay.
a break, Aslam, while we're on that note, just so that you can start again really nicely. Is that fine?
That's fine.
Okay, guys, we're gonna go for a really quick ad break. During this time, maybe just practice your handstands as well, because that was the most awesome thing I've ever seen. See you after the break. Welcome back, Mindsetters. I hope you guys are refreshed. If you have just joined us, we are doing grade 12 life science and we're doing revision today. Revision on some paper 1 and some paper 2 life science. So make sure that your focus is on us today, because these are such important sections, guys. These are sections that we may have not, or may have gone through over the past few weeks. It's always nice to have some revision in your head because it's nice to recap. So make sure at this point you are sending any questions that you may have, any queries, anything. Just shoot it through, guys. We're ready for you. Over to you, Aslam.
Thanks, Katlego. You feel free to stop me at any time if you got some questions coming through, right? Guys, we're still on paper 2. Remember, the ear is part of paper 2. The brain, the ear, the eye, they're all part of paper 2. We are now moving on to the next section in paper 2, and that is the endocrine system. Oops. And there we have a question saying, study the diagram below and answer the questions that follow. I love to put this question on when I'm doing revision, simply because this one gives you the body with all the glands that you need to study. Most of the glands, rather, that you need to study. Some of them are covered in reproduction, so we don't add them in this particular diagram. So the first thing here, remember the levels also, people. In terms of the level of this section, level 1 is to know the names of the glands. Level 2 is to know their position. Level 3 is to know the hormones that each of these glands secrete. Level 4 would be the functions of those hormones. Level 5 would be what would happen if there's too much or too little of those hormones. Level 6 would be to explain a negative feedback mechanism. Now, if you keep these things in your head, then you know that, look, I'm studying this section. What is it going to be when I'm writing the exam? What are they going to require me to do? So I've given you these 6 levels. Level 1, the names of the glands. 2, their position. 3, the hormones they secrete. Level 4, the functions of the hormones. Level 5, too much or too little, what would happen. And level 6, what is a negative feedback mechanism, and give examples of those in the body. So that's what you need to know here. And a diagram like this in the middle of a double folio page, and label all the glands, so you know the position and you're giving their labels. Next to that, you write the function, the hormones that they secrete in another color, and in yet another color, you can write the functions of the hormones. And another color for high, another color for low, for the glands that are tested in the exam. And then you can do a diagram to show the negative feedback mechanism. Let's go to the question. Label the parts numbered 1 and 4. In this case, they're asking for 1 and 4. Let's just do that. I'm hoping I get this right. Okay. 1 and 4. They didn't ask you 1, 2, 4, 1 and 4. So you only had to do 1 and 4. So label. And we always label on the diagram. This one here we call the pituitary gland or hypophysis. These are the only 2 accepted names for this gland. Some old textbooks may talk of the master gland. It is not marked correct at the end of the year, so do not use that word. It is pituitary or hypophysis. And 4, on top of the kidney, these are the adrenal glands. That was the first question. Write down only the number of the gland that, 1, produces the hormone glucagon. So we're going to level 2. What are the hormones? The gland that produces glucagon. And that would be the pancreas, and in this diagram, it is number 3. B, produces a hormone that controls the growth of long bones. Very important here. They're not talking about normal overall growth and development. They're talking about growth of the long bone. And that one is produced by, number 1, the pituitary gland. By the way, that would be the growth hormone. Not the answer to the question, but that's the hormone. Which hormone? The growth hormone. Where is it secreted by? By number 1, which is the pituitary gland. Produces an iodine-containing hormone. That would be thyroxine, and thyroxine is produced by the thyroid gland, and that is number 2. Produces a hormone that is involved in the reabsorption of some salts by the kidneys, and that would be the adrenal gland, number 4. And the hormone there would be aldosterone. Good, guys. We're moving with this question. Not too difficult. State 2 similarities between hormones and nerves with regards to their function. They did not ask you about their structure. They asked you about their functions. How are they similar? They both receive stimuli, and they both respond to stimuli. Both of them need a stimulus. They need to be stimulated somehow to bring about a response. And what do they respond to? They respond to the stimulus. So these are the 2 ways in which they are similar. The next question says, oops, we have to go back, sorry. One more there. State 1 functional difference between the hormones and motor nerves. Motor nerves. How are they functionally different? I'll write the answer here on the top. A motor neuron, first of all, has a specific response in a specific area, whereas the hormones have general effects throughout the body. Just to give you an example, if you take the motor neuron, for example, if I step onto a thorn, I jump away, all right? That jumping, the response that this motor neuron brought about was not in my whole body, but the muscles of the thigh and maybe the quadriceps here that contract and relax to lift my body up, to pull me away. Or if I touch a hot flame, I pull my hand away, the biceps and the triceps are working. So the motor neuron is working on this guy. Or if you walk in the house and you smell good food, the motor neuron will go to the salivary glands to make them secrete saliva. So specific target. Whereas if you take thyroxine, thyroxine is a hormone that controls the heart, it controls the nervous system, it controls growth in certain ways, it controls the metabolic rate throughout the body. The growth hormone throughout the body, not in one particular long bone, but the entire body. So that's the functional difference between them. The next question, two things in one we're going to do here. One is we're going to discuss the answer to this, but in relation to writing an essay. The question is an essay question, as you can see the mark allocation there. Content 17, synthesis 3, and total 20. Remember, guys, synthesis, I was just discussing it before the show with another educator. Synthesis does not mean you have to write a heading down and write something there. These 3 marks, these ones here, synthesis, we give to you regarding the quality of your answer. And life science is not like English or history where you write an essay with an introduction and a conclusion and so on. Life science is a mini essay. We're interested in facts and answering this question. If you answer the question in terms of what they ask you, you should get 2 or 3 marks, depending on the quality of your answer. If you go away and you deviate from this, you're going to get 1 or you're going to get 0, depending how far you deviate. So that's how you get synthesis mark. You do not even write the synthesis heading and write something under it. They say describe the role of the hypothalamus and the adrenal glands in bringing about changes to the blood vessels of the human skin and explain why these changes taking place, or these changes take place. Now, watch here. First of all, they're talking about the hypothalamus and adrenal glands. They're talking about 2 glands. What about them? Changes to the blood vessels of the skin. How do they change? What do they do to bring that about? And explain why these changes take place. So there's 2 sets to this whole thing. In fact, if you really look at it, there's 4 sets to this thing. One is adrenal glands and the skin. How does it bring about the change? The blood vessels in the skin. 2 is hypothalamus and the skin. How does it do it? 3, why does this one do it and why does the other one do it? I would give this answer in the following way. I'll deal with the hypothalamus as a gland. I'll talk about what it does to the skin and how it does that to the skin. And I will deal with the why as well there. And then I will go to the adrenal glands. What does it do to the blood vessel? How does it do it? And why does it do that? And that would be my essay. Let's see if we can get this right. There we go. All right. I'm going to try and write some of the stuff there and some in this area here. First of all, this thing is absolutely sensitive today, Katlego.
Is it working, Aslan?
It's very sensitive.
Yeah.
But it's not sensitive to what I want it to do. But anyway. All right. So we're looking at the hypothalamus first. Hypothalamus. And the skin. I'm not going to write the whole answers, guys. I am just going to do the plan. You will put it together. What does the hypothalamus do? First of all, the hypothalamus is involved in, if you look at the functions, in temperature control. Hypothalamus, temperature control, thirst, hunger, emotions, sleep, appetite. Those are the functions of the hypothalamus. So we are talking about temperature control here. And that's where it comes in to this blood vessel. And there you're going to talk about cold, and hot. On the cold, the blood vessels will constrict. In the hot weather, they will dilate. I'm giving you the plan, you will fit in the other stories. Obviously, they will constrict on a cold day, so less blood goes to the surface of the skin, so therefore less heat goes to the surface of the skin, therefore less heat will be lost by conduction, convection, and radiation. And less blood will go to the sweat glands, therefore the sweat glands become less active, we will sweat less, so less heat is lost by sweating and the evaporation thereof. On a hot day, the blood vessels dilate. And when they dilate, more blood goes to the surface of the skin. And if more blood goes there, more heat goes there. And if more heat goes there, more heat lost by conduction, convection, radiation, and evaporation. And more blood goes to the sweat glands, stimulating the sweat glands to secrete more sweat, more sweating, more evaporation of sweat, more cooling. So that's the hypothalamus. Then we go to the adrenal glands. Remember, the adrenal glands secrete adrenaline, which is the flight or fright hormone. In other words, it prepares the body for emergencies. So in an emergency situation, adrenal glands will secrete adrenaline. By the way, this is a nervous system control. This one here is an endocrine control. Adrenaline prepares the body for emergency. And one, we're not going to go into the whole detail. We are worried about what does it do there. It causes the blood vessels of the skin to constrict. And at the same time, it causes the blood vessels to the digestive system to also constrict. By the way, why? Because these are non-essential parts of the body in terms of an emergency. You do not need to deal with the skin. You do not need to digest food at this time. That lion is behind you, Katlejo. Do you need to digest food at this time?
No way.
No. If you had some food, you're going to leave it and you're going to run. So who's important in this case? The brain is important. Yes. The muscles are important. They considered to be essential organs in this time. Why? Because they're going to react to the emergency. So more blood is diverted to those parts that need to respond in this time. And that is why, the why of this question. I'm hoping you're with me on that one. But you will answer this in an essay nice and neatly. You will do hypothalamus, cold, hot, and explain and all the stories that go with it, and the adrenal gland and adrenaline and how it does the work as well. Any questions we're getting there?
Not yet.
Because we're pausing there. That is question paper 2 that we have covered. We're moving over to a small question on paper 1 that I've been dying to put up for the last couple of times on DNA. I tried it on genetics and we just didn't get to it. So today we're making sure that we got there. The diagram represents DNA profiles from a murder case. So there was a murder, and there we have the DNA profiles, or the DNA sequence, or the DNA fingerprint of the case. This is of the victim. This is suspect 1, that's suspect 2, and that's the evidence, whatever they found, whether it was hair, whether it was blood, whatever, skin, nails, a cigarette butt, whatever they found there, and they took some evidence from there, and they got this barcode, as I might put it. Now, look at this. In your day and age, guys, where the barcodes are such an important thing in our life, you go to a supermarket, a shop, anywhere, Katlejo, what do they scan?
The barcode.
The barcode. And when you go to the till, if they scan a bar 1, does it show lunch bar there?
No, it does not.
It does not, because they are unique. Each barcode is unique. The same way we have our own fingerprint, DNA fingerprint in this way, or DNA profile. They say, which one of the suspects could have been the murderer? We need to look at the evidence. This was the evidence. That's the victim, and these are the suspects. All we're doing here is a matching exercise. That one matches with that one, so our answer would be suspect 1. Back. Explain why a sample would be taken from the victim as well as the suspects. Why do we need a sample from the victim? If this victim was dead, for example, and we did not take a sample from her body or his body. Then, if we took this and this was the evidence, now we bury this victim, or we cremate the victim, or whatever we do with it. Now we're sitting with this evidence. What if the evidence we're sitting with belonged to the victim? We'll be searching for somebody that's gone.
Yes.
All right? So the reason, and this answer will never change, the reason why we take the victims is to make sure that the evidence sample does not belong to the victim.
Because he couldn't have killed himself in that point. When
You're
there's
watching a lot of CSI, huh?
Yeah.
There we go. All these things helping now, right, guys? So that is why we take the sample from the victim as well. So this is how you answer DNA. And remember here, they could have asked you for a reason as well. Why do you say suspect 1? Because suspect 1 DNA matches exactly the DNA of the evidence. That's your answer. No matter how they ask this question, which one do you choose? You choose suspect A or B or Z. Why do you choose that one? Because his DNA matches that of the evidence.
Okay.
And with that, guys, over to you,
Over
Katlego.
for a break, eh? Guys, let's take a quick break. I can already see there's lots of activity on the page. So please keep it running. And if you have questions on what Aslam has done up to now, please send them through right now, guys. I'll be waiting. See you after the break. Welcome back, Mindsetters. It has been such an awesome show so far, and I can see that already by the questions that you guys have to ask, as I've already said. We are going to go through a question right now. This question is by Ntando Mbele, and she says this is the prelim question from the Gauteng paper. So in case you have it, maybe just flip that one over, or if you don't have it and you're not from Gauteng, don't worry, because I'll be reading the question out for you. And it says that it's for 20 marks, Aslam.
Mm-hmm.
Okay, so she says, the question starts, read the statement on population growth forms and answer the question that follows. The characteristic pattern of increase in a population is referred to as its growth form. Discuss the two basic patterns of growth forms and explain why they differ from each other.
Great, Ntando.
Ntando, yes.
Ntando, that was the essay question in the Gauteng paper 2. And this one here needed your knowledge of population dynamics, and more specifically, your knowledge on population growth forms, as the question has directed you to the two basic growth forms. And in the population studies that we do, we talk about two growth forms, and they are, one is the geometric growth, and the other one is the logistic growth curve. So basically, the examiner wanted you to explain these two growth forms. The first one is easy. If you look at the graph, you're looking at a J curve, geometric J curve. Basically, there are two parts to it. There, let's get another color there for now, there, and there. You notice here the growth is not very high, and there the growth is very high. It's almost doubling all the time. So we call this exponential growth. Here we call this the lag phase, and we call this the log phase. Why lag? Because this growth is slow. Why? Because the organisms may not be used to the environment, or maybe they're not sexually mature. After a while, you see it just shoots up. By the way, the human population at the moment is said to be growing on that type of a curve, exponential growth or geometric growth. So you needed to explain this. And to cite an example, you look at compound interest. Because you deposit some money in the bank, you get interest on that, and then you get interest upon the money you've saved plus the interest you have. So it's called compound interest. So it's compounding all the time. It's getting bigger all the time. And that's what we're talking about here. And in the logistic growth curve, this one is a little more complicated in the phases. You're going to get what we call an S-shaped curve. And here, we can see a couple of different areas here. So we're going to use green now here. That would be the log phase. And you're going to explain the log phase. Then you will talk about the lag phase, sorry. Lag phase. Don't get that wrong. That's the lag phase. Okay. This one here is the log phase. some books talk about this part here, just where the growth is slightly tapering off as the decelerating. And this one here as the stationary or equilibrium. Sometimes you may even get a line going down, and that would be the death phase. So you can see, obviously, in this question, you would have got more marks for explaining the second type of growth, the logistic, because there. You're going to say the name. You're going to get a 1 mark to say the name. You're going to explain the growth is slow here, and you're going to explain why it is slow there. Then you're going to go here. You're going to say that the growth, this is the log phase, growth is rapid. Why? Because the organisms are used to the environment or they are sexually mature. Then you're going to say the decelerating because, and we can put arrows there to show that environmental resistance is coming in. That means the environment now can't handle more than this. And sometimes you'll see a dotted line going across there. This dotted line is known as the carrying capacity of this environment. By the way, a definition there, carrying capacity is the maximum number of individuals of a particular species that a particular habitat may withstand, may support. So that's what's happening. The environment starts resisting, and therefore the growth starts flattening out, and you get your stationary where the growth is constant now. It's not shooting up anymore. And if something happens to the environment or these organisms exploit the environment too much, then the environment's carrying capacity can decrease, and then we can have what we call the death phase. So you needed to explain all of that. And in your explanation, you would have then highlighted the difference between these two growth forms. Another question?
There was another question by Munaka. Munaka says, wait, sorry, I'm just reloading the page. Okay, so Munaka says, yes. She says, Aslam, can you please explain more about the interaction that occurs between the hypophysis and the thyroid gland, not the pituitary? She said she made a mistake.
Okay, hypophysis and pituitary are the same gland. That's the first thing. It's the same gland, different names. Hypophysis and the thyroid gland, we talk of what we have there is a negative feedback. And when we're looking at that, if the level of thyroxine, which is the hormone that comes from the thyroid gland, if it is high, there's too much of it in the body. When we say high, we mean above the norm. If the norm is 5 and it's 7, 8, 9, 10, then it is high. So what happens then? Then the pituitary gland, now this is where the relationship comes in, secretes less TSH. And TSH, for the benefit of those who don't know their work, thyroid stimulating hormone. Listen to the words, thyroid stimulating hormone. So what does it do? It stimulates the thyroid gland. And what's the function of the thyroid gland? To secrete thyroxine. So the pituitary gland then secretes less of this. And if there's less of that, then the thyroid gland is less stimulated. And if it is less stimulated, it will secrete less thyroxine. So therefore, the thyroxine level comes back to normal. And we can go the reverse, thyroxine, if there's too little, then pituitary gland will secrete more TSH, more stimulation, more thyroxine back to normal. Your answer must end up with back to normal, because the purpose of this whole exercise is to bring it back to normal. And why is it a negative feedback mechanism? What was the stimulus? The stimulus was too much thyroxine. What is the response? The response decreased the thyroxine levels. So it is a negative feedback mechanism. If there was too little, you'll find that the response is there's more thyroxine in that way. And remember, guys, be very careful here. Most people, they gamble that they'll only ask this negative feedback. In the endocrine system, most of them work on a negative feedback mechanism. If you look at ADH and water osmoregulation, when there's high water, there's low water, negative feedback mechanism. Glucose and insulin, high, low, negative feedback mechanism. So they can ask you about a negative feedback mechanism with almost every hormone. So be very careful. The idea is understand the principle. When there's more of it, less will be secreted, and you must look at the control. For everything, there's a control center. In this case, the pituitary gland is the control center. It gets this information, and it then sends out information to correct the situation. Remember that. We're looking at another question very quickly. No more questions, right?
Yes, no more. For now.
We'll come back to that.
Okay.
The flow diagram shows how bacteria can be genetically engineered to produce human insulin. Sorry, guys, that I'm cutting a bit of the questions. We'll go back to them. The reason being that this question also has been eluding me all this time. Every time we want to discuss this question, we run out of time. Today we have the time, so we've done the DNA fingerprinting. Now we are looking at still biotechnology. The flow diagram shows how bacteria can be genetically engineered to produce human insulin. So how we can change bacteria to give us human insulin. So they're giving you a bacterium A, a ring of genetic material. You see there's a ring of genetic material here. There something has happened. We've cut some part out, and we take a human insulin gene. And as you can see from this diagram, this has been inserted there. And then we have this. We put this in bacterium B, and we allow bacterium B to reproduce asexually, binary fission. That means it's going to make copies of itself, and they are all going to be identical, no change of gametes, no meiosis, therefore no variation. So this, unless mutation takes place, but in most cases they will be the same. That is the crux of this whole thing here. Suggest how the ring of genetic material is cut open. Here's the genetic material. How did it get cut open? And here we say we use here restriction enzyme to cut the enzyme, genetic material. So if we look at it, and I'm saying this with caution. I'm saying this with caution because I don't want you to write this in an exam. We can see the restriction enzyme to be like a scissor that is cutting this thing here. And remember, it's not a physical thing, it's not a mechanical thing. It's a chemical reaction. This is a chemical reaction. This is an enzyme. It's a chemical that causes this thing to be cut. And when it's cut, we then reinsert the human insulin gene into that spot that was occupied by a bacterial gene before. And then we insert this in another bacterium, and we allow that bacteria to reproduce. Very technological stuff, guys, but this stuff is saving lives. Let's look at the questions more. Where is the human insulin gene obtained from? And I'll smack you silly if you say from the human. That's not the answer, Katlego.
Yes, because it says human
exactly.
insulin.
Yes. So where is it coming from? The human where? From the human pancreas. Remember, this is also linked to the human genome. We have identified the different chromosomes, the 46 chromosomes we have. Scientists have identified all the genes or most of the genes on these 46 chromosomes, so they know where in the pancreas this insulin gene will be found. If we didn't have this map, we couldn't find it. This is called the Human Genome Project. Human Genome maps out all the genes in the human body. Wow. So in that way, when we want to fix something, we can start experimenting with some of those stuff. All right? So from the human pancreas. Describe the advantages of obtaining insulin in this way. Now, obviously, to answer this question, you need to know how we got insulin in the past. And we got insulin in the past from the pancreas of pigs, cows, sheep, and other animals. So now we need to look at what are the disadvantages of that as opposed to now. So that's how we get this advantage. First of all, obviously, by telling you pig, cow, cattle, sheep, whatever, you know we have to kill animals to do this. So in this way, large-scale killing of animals is reduced.
Yes.
It's reduced. That's the first one. Yes. Next thing, because it's human stuff that we're using, then there would be fewer or no side effects. Because we're not taking something else. We are using the human stuff.
Yes.
So it's going to do what it's supposed to do. Thirdly, from a religious point of view, there would be no objections now. because we're using human insulin and we're not using from animals. For example, myself, I can't use anything or eat from or use any product that comes from pork or from a pig. So now, if they take insulin from the pig, I can't use it.
You can't
so
use
I'm
it,
disadvantaged.
yes.
So because of that, that doesn't happen now. So these are your 3 advantages. Also, because we're getting it and we're producing it synthetically now, you can buy a vial.
Yes.
Keep it in your fridge. Whenever you need it, you take it and you inject yourself with the insulin. So that also, you can get the insulin on demand. You buy it from the pharmacy, you store it in the fridge, and you use it when you need to use it. Guys, we move on to just one more question today. A group of grade 12 learners were asked to test the following hypothesis with regard to phenotypes. At each age group, boys are taller than girls. This is a hypothesis. At each age group, boys are taller than girls. So the thing is, taller is the relationship. What are we comparing? Boys and girls at different age groups. And we're saying that they are taller. They say, name any 3 steps in the planning process that must be considered in an investigation. Now, guys, whether you are planning the matric farewell, whether you are planning a wedding, whether you're planning an investigation, there are certain basic steps you must do. These steps are called planning before the event. You can't say you must walk down the aisle in a marriage, or you must be able to give out tokens at a matric farewell. The planning must talk about what you do before that to plan this event. And in this case, if we are taking this here, we want to test this hypothesis, we must first decide who is going to do what in your group, 1. 2, you must decide on the sample that you're going to use. What sample are you going to, where you're going to use this sample. 3, you must also look at the size of the sample. You must also look at what instrument you're going to use to do this investigation. You're going to also talk about how you're going to record your information and how you're going to present it. Do not confuse it with actually doing the experiment, because this will get you no marks. If you look at the Gauteng paper, paper 1 asked for planning. Most learners spoke about doing the investigation. They didn't talk about the planning that goes before that. Planning is before the event. Provide a title for the graph. It's showing the investigation here. Obviously, average height in boys and girls in the grade 12 class. At, let's see, yeah, or not in the grade 12 class, in whichever class, at so much at different ages. So notice you have the independent variable and the dependent variable. The height at different ages is measured in this particular graph. Do that, and you have your title there. In your own graphs, you must have that as well. Guys, that brings me to the end of my time today. I hand you over to Katlego.
Thank you, Aslam. So, guys, thank you so much for such a great show. This is a very important show. I hope you guys have realized that all the stuff you have seen in your prelims and you will see in your finals. So make sure that you have actually grasped what went on. And if not, you can carry on and ask your peers on the page. There's no problem. And remember, Facebook doesn't close, guys. So keep chatting to each other. Keep telling each other whatever you need to tell each other and answer each other as well. Also, I want to remind you guys about the spring school next week. It starts on the 1st of October, guys. Make sure that you have downloaded the booklets, you have called to order or emailed to order, because they are going to be very important next week when you do spring school. And spring school is always important, guys. So I'm looking forward to seeing you guys there. From us, Aslam and I, bye-bye, guys. Good night.